Thank you very much, both of you. It's really an honor to be invited here. And, I've experienced sitting on that side quite a bit when I was much younger, and I'm always very nervous of being on this side because I know what you're all thinking. Anyway, but thank you again for having me here. My current job involves scientific policy and advice and working on missions across the scientific spectrum. But what I'll talk to you today is a relatively narrow part, not substantially related to that, but what I think as medical students, the opportunities you may have in today's world to do research in the life sciences. And really, this is not to say that medical doctors must do research, but to actually say something slightly deeper, saying that much of our activities as humans, and particularly as humans who have been fortunate to have a science or medical or engineering or philosophy or history or any kind of education, much of our activities in our days involves questioning what's happening around us, and then using the results we see from that questioning to change our lives or to change other people's lives. So it's not that we can escape from research even if we are not formally associated with it. Research should become an integral component of our lives. That said, much of the great advances in our understanding of the world have come from people whose principal and primary training have not necessarily been in the sciences, but have moved from areas such as medicine or engineering or even the humanities into sciences. So there's an expectation that each year the numbers of people who come to do their MBBS, amongst them, there will be some who actually dedicate their lives substantially to research. And amongst medical scientists, the All India Institute of Medical Science is unusual in the significant amount of research activity which happens here, both in the clinical and in the non-clinical departments. And indeed, the Christian Medical College, Vellore, AIIMS, the PGI Chandigarh, JIPMER, the Sanjay Gandhi Institute in Lucknow, these are the handful of places where a substantial amount of clinical research goes on in the country. Yet, keep in mind that one major American university, such as the University of Texas Southwestern Medical Center or the University of California, San Francisco, has more clinical research going on than all of India combined. So, both in terms of research in the life sciences in general and in clinical research, India affords not only a huge opportunity, which we must grasp, but a huge demand, which we'll be failing to do. So with that, let's go on to see what the life sciences has to offer. And decades, indeed centuries of research in the life sciences has led to an extraordinary understanding of what we can call nature's engineering. So we have, for time immemorial, humans have questioned how they came about to be, how life in the universe and the universe itself came about. And in asking these questions and devising tools to answer these questions, we've got an enormous understanding on one hand of what nature's engineering is about, how all of us and life on this planet and indeed the planet itself has been constructed. But in doing so, we've done two things. We've created new frontiers of questioning, and we've also begun to engineer nature. Our ability to engineer nature distinguishes us from other animals on this planet. And indeed, the scale at which we have done that, and the scale at which we do that through using tools such as language and culture, means that today we have brought upon the planet a pace of change which is unprecedented ever before. All other animals on the planet aim to survive and pass on their genes from generation to generation. But we have transcended that, at least in significant measure, by passing on our learning, our knowledge through cultural evolution and cultural transmission in a manner which will also change the planet enormously. So, through our engineering, we have had buildings, cities, aircraft, ships, language, and so on and so forth, computers, but we've also warmed the planet up, and we've also created huge environmental challenges. So we have not just as medical doctors need to deal with the consequences of our lifestyle and our genes, but also we have to take a holistic view of how the environment and climate change address upon these questions. So in this very pressing point in Earth's history, where we not only need to ask about how we survive, but what we can do for the planet and change the planet back, put it back on track, what are the questions which biologists ask? And this was answered by Sidney Brenner, who died only a few days ago, famous biologist. And he said that biologists ask only three questions. How does it work? That is physiology. How is it built? That's developmental biology, embryology as it was called. And how did it get that way? Evolution. As medical doctors, you ask two other questions. How do you fix it when it's broke? And as engineers, synthetic biologists, you can also engineer nature today because of our understanding. But these are the three major questions which all of us ask, and everything else is a consequence of it. So, when people started asking how it works, and how is it built, physiology and developmental biology, they had all sorts of ideas. I mean, people imagined that they could see a homunculus in the head of a sperm, and therefore argued it was in the head of the sperm and not on the egg. But they argued that this is the way you have one human coming from another human. Of course, it didn't explain how the homunculus came in the sperm in the first place. So there were many of these kinds of questions, but a mechanistic approach to studying embryology, physiology, and so on came about much later. And these are traditions which are there very strongly in multiple civilizations. And all of them ask questions about how the world came about to be, plants, animals, and how they function. And there were many important insights coming from observation, just as in astronomy, but also from some actual interventions. And let me give you two examples of how observation first and then actual interventions has transformed our understanding of life. And that goes to the third question, which is here, how did it get that way? That's about evolution and then genetics. These two people, Charles Darwin and Alfred Russel Wallace, with their theory of evolution by natural selection, completely transformed our understanding of life. Till that time, it was still quite evident that many animals were related to each other. Before Darwin, if you went to the Galapagos Islands and you saw a huge lizard, and you saw another lizard in your home in Delhi or Chennai or wherever, you would have actually made the leap to say that lizards are related, even though one lizard was about 9 feet long and the other was 9 inches long or less. But Darwin made an incredible leap by looking at observing the whole world, and Wallace did that in Asia. Darwin did that in many parts of the world, South America, other parts of the world. What they concluded by looking at big data was not just that all life on this planet was related, but there was a systematic way by which this relationship could be extracted, and there was a mechanism underlying that, and they didn't know the mechanism, but they suggested that natural selection was the way by which speciation occurred. And you all know about Darwin and the theory of natural selection. Basically, all life on Earth started at this wall as single cellular organisms as the first approximation, and accident upon accident and selection in the environment resulted in a variety of forms of different complexities over evolutionary time, hundreds of millions of years. This huge variation came about, but this huge variation had to follow certain specific rules. And it was not unbounded in the kinds of rules which were to be followed. What were those rules? Those rules were rules of genetics, which another person, Gregor Mendel, first formalized. And unlike Darwin and Wallace, he wasn't a big data person. Mendel worked in his garden. He was an experimentalist, looking at small data, as it were, and he cleared through the confusion of various kinds of measurements of size and shape and so on and so forth, and elucidated the rules by which variation took place from generation to generation. So Darwin told us how variation took place and created different species, and Mendel told us how variation, the rules by which variation took place from generation to generation within a species. Now, as I said, these principles of evolution and genetics didn't tell us what was the molecular mechanism, in today's terms, of how these changes took place. Now, the understanding of those came from a very different area, and that area is called embryology or developmental biology. And embryologists did something very different from what these big data people did or what Mendel did. Mendel made crosses and observed phenotypes. What embryologists did was to take embryos, typically amphibian embryos, whose development one could watch in a petri dish, and asked what would happen if you took an embryo and put a hot needle and ablated one part of the embryo, developing embryo, or you cut out a piece of an embryo and put it in another place. So Willem Roos took an embryo, a tadpole at a 2-cell stage, ablated one cell with a hot needle, and asked what would happen, what kind of a tadpole would develop. And he concluded that when he saw that you had half a tadpole developing, that each of these 2-cell stage had the information to make one half of the animal, and therefore the way development took place was by something in the first cell, in the fertilized egg, dividing itself asymmetrically into the 2-cell stage and the 4-cell and the 8-cell and so on. And these division of these components resulted in these cells having specific properties, making different parts of the organism. So this homunculus view which you had, he gave an apparent explanation of how this could take place. But if you, instead of ablating the embryo, what Driesch did in an experiment in Trieste, he took another organism, and much of research happens by repeating other people's experiments in other contexts. He took a sea urchin embryo and at the 2-cell stage put a hair around it and separated these 2 cells. If Willem Roos was right, then each of these cells should make half a sea urchin. Instead, each of these cells made a complete sea urchin. And this was really puzzling. How did this cell know that it was no longer attached to another cell and it could make a complete sea urchin? Driesch was so shocked and puzzled by this research that he gave up biology and took to religion. So there's a danger in experimental science. But this conundrum of how these two differences happen was solved over many decades of work by many people. But there are many kinds of signals which tell embryos how to develop. Roos was right. Cells do partition components to their daughter cells and therefore become different. But cells also talk to each other and therefore know how, whether one cell is there or not and what kind of cell is there. So they're signaling the environment of a cell also matters, not just its parents. And a joke I'd like to say, again modified from Sidney Brenner's joke, is that Brenner pointed out that the first model of cells inheriting their properties is the English model, where you are what you are according to your parents. And the other model where your fate is decided by your environment is the American model, where you stay determines what you are. If you are a big shot in a White House, you're likely to be president. But if you leave the White House, you're no longer president. Yeah? So what you are depends on your environment. Another form of communication is I jokingly call the Indian model, where everyone is equal, but someone randomly pops up with a different fate, such as in notch-mediated signaling, and suppresses everyone else. So there are multiple ways of signaling which take place, and these result in differential cell fates. A very interesting way of signaling, though, is a long-range signaling mechanism where groups of cells in one context, send out long-range signals to a population. And the population responds to that depending on what their parentage is or what their previous history is. So therefore, a given long-range signal is interpreted by different parts in different ways, different tissues in different ways. So these kinds of signaling basically allow the use of a relatively small toolkit of molecules and mechanisms in multiple contexts. You use the same screwdriver, spanner, rivet, and so on and so forth. But because history matters and context matters, you end up making different kinds of structures. And that's why you have, at the higher regulatory level, shared components connected by the thread of evolution and by the thread of DNA, that these chemistries are consistent across all organisms, but are used in different contexts in different ways. One very nice example of that is the Spearman and Mangold experiment, where Hans Spearman and Hilde Mangold, who sadly died in a kitchen stove accident and therefore didn't get the Nobel Prize which Spearman got, what they did was to do this experiment and ask, if here's a group of cells which is normally part of one part of the organism, what would happen if I take this and put it far away in another location, right? Would those cells over there behave like this and differentiate like this? Would they behave like those cells and differentiate like that, according to the two models we've discussed? But what they found was something amazing. These cells over there created a new ecosystem, a very small number of cells. They remodeled the ecosystem where they were and changed the properties of those cells. These were organizer cells. And here is that's Eddie D. Robertus, professor at the University of California, Los Angeles, repeating the Spearman and Mangold experiment. He's taking out a piece of tissue from one part of an embryo, right, with very sophisticated tools, as you can see. And then he's taking this and pasting it, tapping it onto another part of a different part, not the place where he took it out from, but another place of a host embryo. Then he goes home for his supper and comes back and sees what happens. And lo and behold, he gets a tadpole with two axes developed, with two heads, moving around. And this is absolutely stunning and amazing. And this is what Hans Spearman got the Nobel Prize for. The proposed explanation for this is that some chemical, the organizer, secreted from one region which organizes the other. And people searched for many years using biochemistry for these kinds of organizers with very poor luck. But once genetics came about, they could actually find out what these kinds of molecules were there in much better ways. Which then takes us to genetics. As I told you, Mendel and Darwin told us how, or at least told us in theory how variation could take place and create species, or variation from generation to generation. What is the underlying mechanism, and what has transformed this from a theory to actual fact? Why is evolution no longer a mere theory, but is absolutely in proof, right? And how do we know how variation takes place from generation to generation? Those kinds of studies were led by people such as Thomas Hunt Morgan and his students Alfred Sturtevant, Calvin Bridges, and others, who looked at characters in adult flies, phenotypes, and looked at variations in those characters and saw how those variations were inherited from generation to generation, and whether those characters went together and how they went together. So they could show that all these characters they studied could be put on essentially 4 sticks, as they were, on 4 imaginary sticks, and depending on how far they were from these sticks across from top to bottom on that diagram, they were either very likely to go together from generation to generation or less and less likely. And those which mapped onto another stick segregated much more independently. So this was their insight into the functioning of an organism without ever actually looking inside. They're proposing that characters are coded in today's language or on something inside the animal, and something inside the animal is organized in the following manner. And Morgan could actually see quality in chromosomes in Drosophila, and because of work by Hermann Muller and others, came to the conclusion that chromosomes were the site where genetic information was encoded. Much later, Avery, McLeod, and McCarty, and then Martha Chase and Alfred Hershey, in different ways, showed that DNA is the genetic material. Right? And therefore, when people found DNA was on chromosomes, they knew that somehow DNA is encoding the DNA has the information for encoding characters. But how does that happen? That information came from work by Francis Crick and Jim Watson, using data from the King's College group. Rosalind Franklin and Maurice Wilkins are shown here. And this allowed us to know the structure of DNA, not only the structure itself, but how the double helical structure allows information to be encoded. That was followed by work led by Crick and Brenner and several others, which allowed us to understand how information in DNA could be translated through mRNA to make proteins, and how proteins are collinear with this genetic sequence, and how that can result in function of molecules themselves. So that's all very well, but how do we integrate all this into functioning of a structure, for example, as complex as a brain? This is the fruit fly's brain. Over many decades of studies, 2 things happened which completely transformed our understanding of biology. First of all, people in organisms such as fruit flies continued to make mutations and understand and link those to phenotypes. But this didn't tell us what was going inside the organism. But once DNA sequencing became possible and the isolation and cloning of genes could be possible, people could isolate regions of DNA encoded by these mutant genes and look in wild-type animals where the wild-type gene was. But a major change came with our ability to take genes from one organism and put it in another. So if you had a mutant fruit fly, and if by DNA sequencing you found that it had a particular sequence, you could take that wild-type gene and rescue the mutant phenotype. But you could also take that wild-type sequence and look for homologs in other animals, mouse, human, elephant, donkey, and so on. And you could take those human homologs, right, which you did not by the way you do today by bioinformatics, but by actual experiments. You could identify those homologs and you could take them and put them into fruit flies. And amazingly, you found whether it was cell cycle or body patterning, these genes from human could rescue the fruit fly phenotypes. This is absolutely stunning, showing as I said earlier, that the toolkit which you use to make a fly or a worm is pretty much the same toolkit which you use to make a human. What then is the difference between human and fly if the toolkit is the same? It's the same as the difference between a small Cessna aircraft and a Boeing. You use the toolkit, but there's an instruction sequence which you have to follow about what tools to use where, what size wing to make, what kind of engine to put, how many passenger seats to put, and so on and so forth. And that results in a very different organism. Following a set of rules, you can make stem cells make one part of the brain, and if you change the rules, you can make it make another part of the brain, and so on and so forth. But these same principles of how you apply the toolkit also apply, and these principles, the instructions, as it were, need to be interpreted at the regulatory side of genes and not at the structural side. So the interaction of the regulatory side with the environment, with the cellular environment, is what determines what genes come on when and what genes go off when. And therefore that complex set of information between environment and regulatory sequences are what makes for different animals. So this has all been done in very laborious and slow ways. This has been done, is there a clock here? How am I doing for time? Not too bad, okay. I'll finish soon. This has been done in ways which have required individual scientists to study individual genes over many, many years. Hundreds and thousands of such scientists, understanding the expression patterns of genes and what those genes encode, and taking them and putting them back in different organisms and looking at function. And all this today has changed dramatically and moved at warp speed because of technology. So genetics is dominated by a partnership with genomics today. The imaging tools of microscopy and imaging of various kinds, MRI and so on and so forth, have transformed what we see inside the cell. Spectroscopy has changed dramatically. Structural biology is revealing information about the shapes and functions of molecules at much higher speed than ever before. But given all of this, our question is, how can this help understand biology better? What are the questions? And how can we understand biology and apply biology better? Now the problem we have is that the ease of getting information has dulled us into a view that somehow the very act of getting that information and looking at that information somehow is understanding. The biggest challenge in biology has been for a long time, and will continue to be for some time, at the level of the cell, the gap between genotype and phenotype. How does the cellular phenotype come about as a consequence of our genotype? And then how groups of cells interact to give rise to tissues, and how they interact in other contexts such as the brain to result in behavior. So these big challenges remain, and are they going to be, is our understanding going to be improved by better and better collection of data? So people say that there are enormous promises of data, and others caution about the perils of big data. And Sidney Brenner pointed out that technology gives us data across scales very rapidly, but is more better. And across scales is something very important in biology, because biology, unlike the physical sciences, acts immediately across scales. A photon falling on my receptors in my eye results in my responding to something, throwing a ball or catching it and so on and so forth, at very high speed. So right from the level of a photon to the level of an organism, things act in biology very rapidly. That's not typically how they act in physics, for example. You can divide our physical principles and laws into quantum and Newtonian and so on and so forth, and in different ways. Relativity is not something which is important in our day-to-day lives usually. So this kind of partitioning is something which we can readily do in physics, but it's not it's not the way our bodies actually function. We function simultaneously across many scales. So in that situation, how do we understand biology? So the sea of big data today goes across from the nanoscale to the system. There are extraordinary successes of big data in understanding disease phenotypes, in association studies of genes and phenotypes and so on. But is it just that these victories have picked only the low-hanging fruit and things will be much more complex from there? How do we go on from there? And this is particularly of importance in today's world, where we are told, and you will be told as medical doctors, that pathologists will be dispensable because by analyzing images from every possible sample and having millions of samples as your learning set, you can actually make predictions about anything. Similarly, biochemistry in a detailed level will predict disease phenotypes, and genetics will tell you, genomics will tell you about association studies which are inherited. So, is this true? Is this what is going to happen? And if so, what is the point in understanding the principles by which disease is caused or the biological basis of disease? Because you can actually look at phenotypes and make predictions. Who cares about how things work? If I can say that this data at this time equal to 0 moves to this data at t equal to 1, and by looking at millions of samples of all sorts of variations here, I can predict all sorts of variations over there. I don't really want to understand how one kind of signaling mechanism resulted in that and so on. So are we going to escape from the need to understand the basics of biology and go into a data-driven conclusion? So big data can be extraordinarily valuable, as I said, and you'll find many people, particularly with gray hair, bemoaning the onslaught of big data and saying how great the good old days were when you could actually think about a hypothesis and do something, as opposed to today where everyone sits on a computer and comes to conclusions. That they'll say that young people today don't think, they just blindly follow data, and all of that is completely wrong. So big data is extraordinarily valuable, but there are many challenges. And again, these challenges were very well amplified by Sidney Brenner. And Brenner, one must remember, was an extraordinarily intelligent person, very thoughtful, but he also had the ability, and he could cut to the chase, but he also had the ability to change his mind when confronted by data. So he was remarkable in, changing his opinions, but not changing the fact that he was often right, always right. So Brenner points out that we need today a theoretical framework to understand big data. It's not sufficient for us to get a constant, huge bit of information about what's happening at the flyover near you, right? Just having a photograph of every single car, scooter, bus which goes over there is not necessarily going to give you an understanding of traffic management. That in itself is not enough. You need to understand people, sociology, cities, economies, a lot of other things which result in that big data movement. When you do all of that together, along with that big data, you will understand with your engineering knowledge and other knowledge what to do over traffic. And similarly, that's the same thing with biology. He also points out that although we may believe that more is better, getting more data is better, history has told us across biology, and it may be changing now, but still we see again and again examples where least is best. The discovery of CRISPR-Cas9 mechanisms, for example, from studies in bacteria, show us again how inquisitive approaches can be extraordinarily valuable and transformative. Which is not to say big data is not enough. Refining those have involved the using of big data hugely, but the primary discoveries were not based on big data, in that case at least. So we need both theory and a firm grasp of nature of the objects we study to predict the rest. If we don't have this firm grasp, we talk only about artificial intelligence, but we don't have real intelligence, we have a slight problem. So we have to combine your real intelligence in each domain, as physicists, biologists, engineers, and so on, along with artificial intelligence. That intersection can give you a great success. So in this article, Brenner talked about how data alone does not speak for biology. And he points out that generating data is very different from understanding biology. The problem today is you go to a top place or a middle place or a bottom place and you ask them what they're studying, and they hypothetically will tell you diabetes, and then you ask them what they're doing, and they say we're studying a patient cohort. And you ask them what they're doing, they say they're collecting data, and they'll collect molecular data, mass spec data, genomic data, but you very rarely get an answer of what they're doing with the data. What is the underlying biology? So there's a great danger, which they're questioning, there's a great danger that we will lose the understanding, the questioning through the lens of an understanding of biology and expect somehow the data to be magically speaking to you. In some cases it might, but those are the low-hanging fruit which we talked about. But in most cases, if you address data with a deep biological understanding, you'll get interesting answers. For example, if you as a neurobiologist or a neurophysiologist or a neurologist or a neurosurgeon wanted to understand how the nervous system works, there are two approaches you can take. Today, you would take single neurons and do single cell RNA sequencing. You would sequence the genome of the organism where the neuron came from. You would do mass spec to see all the molecules in the neuron, and then you would sit and stare at this, and you would say, let me now figure out how a neuron works. And you would not be able to figure out how the neuron works. You might even have, all sorts of ways of looking at it. You might do calcium imaging to see how the neuron functions and so on. What you need to do today is an equivalent of what Hodgkin and Huxley did with the giant axon of the squid, where they took the neuron, squeezed out its internal parts, voltage clamped it, set it at different voltages, poked needles into it, and changed the extracellular medium, changed the intracellular iron concentration, and came up with a model of partial differential equations which told us how the action potential propagates. Right? And this has at its foundation the prediction of ion channels, molecules which function in cells in specific ways. Those molecules are hidden in your big data set, right? But they will not stand out just from the big data set. So if you combine your big data today, if this had not been done, with measurements of physiology and do the equivalence of these kinds of experiments, then you would come to an answer. Big data is immensely helpful because having identified the protein molecules functionally in these cells, if you had sequence information, you could actually clone these molecules readily from any organism and from any other organism, and you could actually put them back and look at the function in cells and look at how not only how the action potential is conducted, but how similarly you can study synapse and synaptic function. So to understand biology, we must remember, whether we are doctors or engineers studying biology or biologists studying biology, we must go to the core and you must address the key level of abstraction. What is the level of abstraction you're looking at? Right? And that answer is, once we answer that question, we will be able to answer or address every problem in biology. Today, and for many years now, there is a field of biology which many people are enamored by, systems biology. And systems biology, like the study of big data, is a very important area. I wouldn't for the slightest bit demean it in any manner. But remember that this, again, while being valuable, has a problem. The conversion of data into knowledge is what our challenge is. Take our data from systems approaches. How do we convert that into knowledge as opposed to a compendium of data? Sidney Brenner, for example, again, famously said, both the telephone directory and a Shakespeare play have a fantastic plot of characters, right? But one is the plot, one is a drama. The telephone directory is useless. It doesn't tell you anything about a plot and a play. So you need to convert the data of this telephone directory into a plot and a drama. Right? And that's what we need to do. So systems biology claims to solve this problem. The telephone directory does not solve this problem. And real systems biologists are not just collecting the telephone directory. Just as Darwin, who was one of the world's first systems biologists, didn't just collect the data. He thought about it and he analyzed it, right? So deducing models of function from behavior of a complex system is a very challenging inverse problem, right? There are many, many, many possible solutions to a complex collection of data. Looking at the diversity of life on Earth, it was Darwin's genius that he hit upon the right solution. There were no shortage of people before him who hit upon all possible, theoretically valid, wrong solutions, right? But it is very difficult to have a large set of data and hit upon the right solution going downwards from the data. You need to have an understanding of biology and an understanding of ecology to be able to come to the conclusions Darwin did. And he was a biologist looking at biology in multiple contexts. So he brought his understanding of physiology and ecosystems and biology to the deduction of conclusions from big data. It wasn't just a guess. If you want to make a guess, you can invoke all sorts of principles. That was absolutely amazing. So the inverse problem is impossible to address because, very simply put, the square root of 4 is 2, but it's also minus 2. And when you have, larger sets of numbers, the possible solutions to generate that set are many. But which is the right one? You need to go to the core of it. So, to solve this problem, you can't escape to holistic approaches. People who study the brain like to say, I want to take a holistic approach. Or people who study, tissues want to take a holistic approach. And this sounds very nice. It sounds that I'm studying the whole thing rather than small parts which you silly molecular biologists are studying. Ecosystems, people say, I want to study the ecosystem. Very good, it's necessary. But, you cannot escape from genes because these holistic parts are linked, as I said, very tightly to molecular descriptions which are there in their genes. But genes are not the answer. Since genes are molecules and they exert their function through other molecules, the molecular explanation must constitute the core of our understanding. No question about that. But the molecular explanation cannot come from your telephone directory. It must come from the Shakespeare play. And that is the right level of abstraction to see the components over there to see what happens. So one can solve the forward problem and link it to big data is important here, of computing the behavior of a system from its components. You look at the characters in a play. What are they doing? Why are they doing? What's their background? Where do they come from? Where will they go? And then this big data of the play becomes meaningful and you can understand. You have to look at the whole story. You cannot just look at the telephone directory or the cast of characters and say, this is the story. So the correct level of abstraction for this, like the play, the correct level of abstraction is the cell. It is the cell in all of biology which links to other cells, its own function, gets information from the outside, looks at the information it has in its DNA, links them through, varies its function and so on. So if you want to understand biology, at the center you need to have cells. You need to then link that to groups of cells. You can link it to tissues. You can link it to the behavior of the organism. You can link it downwards to DNA and RNA. But that is your core, and that's what your understanding is going to be. So, let me end by pointing out and going to another person. He died in 1954. He killed himself because he was being persecuted because of his sexual orientation. This is Alan Turing. Alan Turing was a mathematician who worked in Bletchley Park cracking the Nazi war code. Bletchley Park was, halfway between London and Cambridge. So when he wanted to talk to his political masters, He put on his running shoes and ran to London. And when he wanted to have intellectual interactions, he put on his running shoes and ran to Cambridge. So he was a long-distance runner, a very applied person, but one of the greatest fundamental scientists ever. And if there's anyone to inspire us as a role model of doing so-called, fantastic quality basic science, and also using that in very important crisis situations in an application, it's Alan Turing. And he made three kinds of major contributions. He had theories on how patterns in development took place. Why are digits or zebra stripes or fish scales, how they develop, what's the basis for that. He talked about computing machinery and intelligence, that's the brain. How, if one of you is actually secretly a computer and not a human, what is the Turing test you do to detect you and bring you out as a bot and not a human person. And computable numbers, which is the Turing machine, which given enough memory, you can actually compute anything. So the Turing reaction diffusion model is an example of how he studied proposed patterns. And this is a review by Kondo and Miura, a bit outdated, but basically if you have one morphogen and depending on its concentration, if cells have different outputs, you can get 1D or patterns. If you have two morphogens interacting, you can get 2D patterns, you get more complicated patterns. But if you have feedback loops and inhibitory components over there, you can get extraordinary self-organizing wave spots and stripes and a labyrinth and so on and so forth. And there are many examples of that in cell biology, in chemistry, where people have tried to model the pattern seen with potential molecules which could be involved in generating these patterns, such as in snails and so on and so forth, in fish and so on. And there are some real-life examples of these, including these of specific molecules shown here. There's a lot of controversy about whether Turing mechanisms actually do operate in this. There's a huge amount of very exciting cell biology which is happening in the recent past, which point to other interesting mechanisms, but Turing still plays a very important role. Turing also talked about, as I said, computing machinery and intelligence, and he could ask if one of you is secretly an Apple or a PC, and what kind of questions you would ask people in the audience to check whether they're secretly a computer. And if you open up their brain, you would open up their head, you would see wires inside instead of neurons. So this is very important today in the context of our better and better understanding of computer science and our ability to create computers which can actually use big data rapidly to compete with us, such as in games of chess, but potentially also have networks which can have supposedly moved towards consciousness. Finally, Turing also invented the Turing machine, which he conceived as a machine which, given enough memory, can compute anything, and it could actually set the rules for computation. But John von Neumann conceived of a constructor machine to make Turing machines, and using a Turing machine to assemble another machine. And he noted that if you made errors in making these machines, you can actually get something akin to what we call mutations. Therefore, if you could, hypothetically in today's jargon, select for these mutations of value, you basically have a mechanism by which you have a replicating computer and a selection mechanism to choose for function in different contexts. So, the concept of the gene as a symbolic representation of the organism, this is important. There is a code script in the genes, say some people, right, which is a symbolic representation of the entire organism, is a fundamental feature of the living world and must form the kernel of biological theory. This is what Cindy Brenner said about Turing. Now, Erwin Schrödinger, who is shown here in exile in Dublin during the Second World War, where he gave a set of lectures which was transformed into a book called What is Life, which inspired many chemists and others to go into biology. He mixed things up. And Schrödinger's confusion comes from the understanding of the Turing computer. Turing invented the stored program computer, and von Neumann, as I said, showed that the description which is there is separate from the constructor, right? So So you need to have a constructor to make the machine, and you need to have a constructor to make another machine. But this is not what Schrödinger said. Schrödinger conflated these two, the program and the constructor, in what is life. And he said that the chromosome, where Morgan had shown, he was inspired by Morgan's work and Delbruck and Timofey Abrasovsky's work calculating the size of a gene. Schrödinger said that chromosome is both the architect's plan and the builder's craft. So it's something which, you get, but also not only tells you what the architect's plan is, but also tells you what brick to put where, how to put it, when to put it, and so on and so forth. It's not just a general plan. But this is wrong. The code script only contains a description of the executive function. It says, these are the components which need to be in place. It doesn't tell you what you need to put where. And that, the left-hand side problem, the part of the problem, how do you control what you do with the code script, is what is missing. It's not there intrinsically in the genes. It's a gene-environment interaction. Right? And that is something which Schrödinger missed. Hodgkin and Huxley's equations, right, is an example of that. When you ask what is the unit of abstraction. And there you understand how you need to understand the control elements to see how these molecules are put in place. So to understand the constructor part of the machinery, the cell is the right level of abstraction. So, to conclude, biologists ask 3 questions, and you as medical doctors will fix it when it's broke. Today, engineers can engineer nature in a huge way, but the problems we address are embodied in physiology, embryology, and evolution. And at the core of everything are the tapes, the DNA sequences which contain the description on how to build the Turing machine, not the executive function. The executive function is a very complex interaction between genes and environment, which we do. So, Brenner ends his essay by saying biology is in crisis. This was in 2012 and still valid today. But he says that the way out is from within. Too many scientists all over the world think that we can solve our problems by looking at technology and data, which, as he says, are important but not sufficient. So as Indian biologists, what should we do? Can we think of a way through today's challenge? There are enormous opportunities around in India today, and while we must learn from what's happening all over the world, we must also be inspired by the quality of people we have, such as you, and the quality of problems in our environment, to think through big data, but also anchor ourselves in questions in environment, in epidemiology, in health, in disease, to frame questions which go to the fundamentals of biology and ask how we can use data to solve this problem. So in this, there are many, many fearsome gorillas in the room, but for now, let's focus on 2. We constantly talk about fixing the system as if some magical fix somewhere else will solve all our problems. It is true, we need a better functioning institution. We need our institutions to interact with each other. We need more resources. We need less bureaucracy. All these are very good things. But we must distinguish between the answer to a question, the solution to a problem. Answers to questions are simple, right? Institutions need to be better, let's make them better. Solutions to the problem are complex. Just as with evolution, evolution is messy and it's just about satisfactory, right? It is not perfect. Similarly, solutions to our problems will always have to deal with the context, our people, what we already have, what we can do, resources, ability, and so on. So we must embrace approximate and try to excel in the approximate and not imagine that somehow magically something will transform. Right? That's one. Secondly, we must fix ourselves as scientists. We must at least for some time every day, maybe half an hour, maybe 15 minutes, maybe 1 hour, think about questions in science, whether it's our research or whether it's our activities, and understand what is going on in the best places in the world. Today's internet allows you to keep the company of the best people anywhere in the world and use that to address your lifestyle. Finally, this is a lesson which all of us, whether in government or as a group over here or in departments at All India Institute or elsewhere, need to embrace. Just as I showed you a two-headed tadpole, here's a two-headed turtle. We have only one body, but unfortunately the way we function in our departments in the government, in our state governments, in our science institutions, within institutions, is we have many heads. Even though we have one problem, we don't think together as one. We put ourselves in departmental silos, in silos of science, and we barely talk to each other. The All India Institute used to be absolutely terrific when it was in its early stages in collaborating between departments. But it, as all other institutions, has grown to a situation where collaboration is lower than it should be. It's still there. But given the huge pressures of the day and what you need to do, your work and so on, you're dominated by chinta and not enough by chinta. Right? So you must have some time for quality thinking and working together. Either be interdisciplinary in yourself or interact with others, but look at problems and their solutions, and all it takes to solve the problem, and not use mass spectrometry because you're a mass spectrometrist or a genetics because you're a geneticist, but use solutions which are best for solving the problem. Then you can practice using a variety of methods. So, resources, processes are difficult, it's not easy to get, but the principal handicap to doing quality science is our culture of science and our thinking about science. We think more incisively, then with whatever resources we have, as resources increase, it's much better, we will do much better. So these solutions will be midterm, but as intellectuals, as researchers, as people who are very, very privileged in having such an education, we must question ourselves hugely and question everyone else. Thank you very much.